Hi, everybody. My name is Ljubenkov. I'm a member of the clinical trials group here at the Memory and Aging Center. It's led by my mentor, Adam Boxer. And I'm going to try to segue with what you just heard about. You heard about pseudomedicine. I'd like to talk about the other end of the spectrum. Just wanted to mention that I don't have any financial relationships to any of the clinical trials that we're going to be talking about today. And I also want to mention that the contact information for our trials group is right at the bottom in case you want to follow up with any questions. Feel free to reach out to us. Here's a brief outline, but I think in the interest of time, I'm just going to dive right in. First of all, how do you know if a drug works? How do you know if a drug has actually gone through a rigorous process that shows that it's right for the drug that for the disease that you want to treat? There's a few things you have to keep in mind. One of them is something we call the placebo effect. Unfortunately, a biologically inactive drug can actually still make you feel better. The reason for that is, if you expect to feel relief, you could actually subjectively suppress your suffering. You actually have neuroanatomy that's developed that just does this process, that makes you feel better if you think you're going to get better. The other thing I'd like to mention is something we call observer bias. Doctors want their patients to get better. We want to know that if we give you a medication that you're actually going to benefit from it. The thing is that if I give you a medication, I'm really, really hoping that it's going to make you better. I'm actually going to be psychologically primed to see a positive impact in you. So we have these biases built into us. And so if you have a person that you want to treat that has a disease that you're trying to cure, you can't just look at the effect of a drug in that one person. You have to look at a whole group of people receiving that drug. And you have to compare that group of people to another group of people receiving a placebo, a biologically inactive drug. And you have to kind of do it at random. And you have to make sure that both the patients and the doctors don't know who's getting the drug. We call this, this is the gold standard for a clinical trial, a randomized, double-blind, placebo-controlled trial. And this allows you to collect unbiased data and really perform an unbiased analysis of what is actually working. And there's actually phases to these clinical trials. Phase 1 is a relatively small trial. That's really just looking to see, is a drug safe? Is it well tolerated? What dose do we use? And is it even getting into the brain in the case of a neurological trial? Phase 2 is slightly larger, and it's really the first swing at trying to see, is the drug efficacious? But it's still not usually large enough for the FDA to approve a drug. Really what you need is something called a phase 3, which can sometimes be thousands of people. And these are these big multi-center trials. And typically the FDA requires 2 positive phase 3 trials to approve a drug. Now, if you're talking about a very rare neurodegenerative disease, some of these rules might be bent a little bit, but this is usually the landscape of clinical trials. When you're talking about Alzheimer's disease clinical trials, you're usually talking about a story of 2 proteins. Amyloid beta that forms these amyloid beta plaques out around brain cells, and hyperphosphorylated tau, which forms these tangles inside of brain cells. And really for the last decade, the field of Alzheimer's clinical trials has really been obsessed with amyloid beta. So I'm going to get a little bit into the biology and what kind of trials we've been running in amyloid beta. So amyloid beta starts as this protein APP, amyloid precursor protein, on the surface of brain cells. It gets cleaved by gamma and beta secretase into this little piece, and we call a monomer, and these little pieces start sticking together to form fibrils, and that's what's getting laid down in these plaques. And so there have been a lot of different trials trying to target different stages of this biology. Beta and gamma secretase inhibitors, unfortunately, those haven't worked so far. Drugs targeting the individual monomers, unfortunately, those haven't worked so well either. So really, the current state of clinical trials right now is developing these immune proteins, antibodies, to activate the immune system to try to remove these plaques. So a lot of the main Alzheimer's trials right now are kind of focusing in on this method. Now, unfortunately, we've already had some failed trials using this methodology, but some of the drugs using this method that are particularly immunoactive actually have had some interesting results that kind of made a splash in the media recently. So I'll cover some of those. And here's just 2 of them. Now 2 of the drugs that made the biggest splash happen to be involved in trials run by a company called Biogen. One of them you might have heard of is called aducanumab. This is really just phase 1 data, so it's hard to know how to interpret it, but at least in a phase 1 trial, aducanumab seemed to be really good at clearing amyloid beta plaques, so actually removing the amyloid beta plaques from people's brains. So this is a picture of something called a tau PET image, and And so it's basically a picture that a special kind of imaging modality that allows us to image the amyloid plaques in people's brains. And compared to placebo, people that were receiving the highest dose of aducanumab seemed to be able to clear their plaques after 1 year. Now again, I mean, that's a pretty picture and it's really exciting. You still have to show that there's a clinical benefit. We still have to show that we're actually treating people. And since it's a phase 1, it's really still too early to do that. The other thing I want to mention about this trial, it's probably not a trivial point, is that, remember, these are highly immunoactive. We're really trying to use the immune system to remove amyloid beta from the brain. Some of these drugs can actually cause inflammation in the brain, and that was the case with the aducanumab trial. It actually, in the highest dose group, in particular, there was a lot of patients that developed this syndrome we called ARIA. And it seemed to be related to, carrier status of one of these genes that Jamie mentioned earlier, ApoE4, so Alzheimer's risk gene. People who carried that version of the gene were particularly at risk of developing ARIA. So, you know, I mean, patients who stopped the drug actually recovered and did well, but it's something that we're going to have to think about going forward with these types of trials. A similar drug also made a splash in the news recently, BAN2401. Really similar drug. Bottom line is, in terms of the imaging data, it was pretty similar. It seems to clear plaques pretty well. And because this was a phase 2 trial, they were able to show that there was a dose-dependent clinical improvement. This is really exciting. But again, this is a phase 2 trial, so it's still not enough for the FDA to make a conclusive statement about whether or not the drug works. The other thing I want to mention is that because of these issues with ARIA, the government regulators actually had required the sponsors of the trial, once the trial had already started, to start removing people with ApoE4 carrier status from the high-dose group. And that happened to be the group that they showed the biggest effect in. So it's always kind of hard to interpret data that's been curated like that, like once the trial's already started. So the bottom line is that you really need phase 3 trials to prove that these drugs work, and that's exactly what's underway. So there are trials in both of these drugs currently underway, big multi-center trials. Now, I mentioned that a lot of amyloid beta trials have failed so far. Part of that might be because we weren't targeting it in the right way. Part of it might be because it's maybe not the optimum protein to be targeting. Now, amyloid beta, we think it's necessary for Alzheimer's disease to occur, but the burden of amyloid beta doesn't actually correlate with your disease severity. It's actually the burden of tau that correlates with disease severity. And Dr. Rosen had talked about this a little bit. So tau sort of gets started in the brainstem, but by the time people start developing amyloid beta in their brain and start developing more frank Alzheimer's disease, you start to see tau accumulating in the memory centers of the brain, and then it sort of spreads out from there in a staging process that we call Brock staging. And it's really this Brock staging that tightly correlates with what people look like clinically. So we think it might be important to target tau, and maybe it's a better protein to target in Alzheimer's clinical trials. So that's a big focus now in current Alzheimer's clinical trials. So I just want to talk a little bit about tau. So its day job is actually to stabilize these things called microtubules, which are like the skeleton of brain cells. And tau can actually get misfolded, and it goes through a process called hyperphosphorylation. And these little bits can actually coerce their sister proteins to misfold, and it can cause this kind of chain reaction inside the neuron that will form these tangles. And we think those tangles are toxic to brain cells. Those little bits, those misfolded bits, can also spread from brain cell to brain cell, and we think that process is what is a method of which Alzheimer's progresses. So there's a whole bunch of different strategies that have been proposed that everybody's thinking about putting into clinical trials. These are a list of a few of these strategies to try to remove tau tangles. One of these strategies, blocking something called tau acetylation, is the basis for one of the clinical trials we're doing here at the Memory and Aging Center with a drug called Sosilate. We have tried a trial stabilizing microtubules. Unfortunately, that was not well tolerated. What I'll mention is that just like amyloid beta, really the current emphasis in tau clinical trials is really, again, using the immune system, using these artificial antibodies to target the tau proteins and try to remove them from the brain. So these tau immunotherapy trials. And there's actually a lot currently underway in Alzheimer's disease and other diseases that involve tau. And so I just wanted to touch base about one of the trials that we're currently running here at the Memory and Aging Center. Unfortunately, the drug has a very long name. It's a trial run by Eli Lilly. It's a phase 2 trial in Alzheimer's disease. I wanted to mention it because it actually has an interesting inclusion-exclusion criteria and sort of just interesting trial design. So remember, we talked about Brock staging. This trial is actually going to try to target a specific Brock stage. It's really an early adopter of kind of this concept of doing something like this. And they're specifically going to target this moderate stage. They think it might be the optimum stage to intervene in this disease. And the way they're going to do that is by using a tracer called fortalsapir. This is a tracer you can give people intravenously. It'll stick to tau in the brain and allow you to actually take a picture of how much tau a person has in the brain. And through this trial, I mean, they're primarily going to measure clinical endpoints, but through this trial, they're also going to see if this drug is actually able to remove tau from the brain. It's kind of an exciting thing to try. I should mention, Alzheimer's disease is not the only tauopathy. There's actually a lot of other diseases that involve tau, as Dr. Rosen had mentioned, and particularly diseases under the umbrella of frontotemporal lobar degeneration. That includes behavioral variant FTD, primary progressive aphasias, particularly the non-fluent variant, progressive supranuclear palsy, cortical basal degeneration, and genetic tau mutations also involve tau. Traumatic encephalopathy, so when people get head injury and develop dementia after that, that also involves accumulation of tau. So there's a lot of different diseases in which we might want to target tau. But some of these are kind of rare, and so that begs the question, are some of these diseases too rare to really fill a traditionally sized clinical trial? And that begs the follow-up question, is there a way to leverage the similarities between these diseases, the common mechanism, to actually build one clinical trial that unifies a lot of these diseases? And the interesting thing is that's exactly what they do in cancer research. So in cancer research, you may have a lot of superficially distinct diseases, breast cancer, ovarian cancer, that share a common molecular mechanism. And there's a new clinical trial design that's starting to fall in vogue, particularly for phase 1 trials, called the basket trial, in which you enroll a lot of superficially distinct cancers that actually share a common mechanism into one trial. And so at UCSF, we're actually going to do a tau immunotherapy basket trial. But instead of having different kinds of cancers, we're going to enroll, I should say we are enrolling, this trial is actively enrolling right now, cortical basal degeneration, non-fluent variant, primary progressive aphasia, traumatic encephalopathy, and tau mutation carriers. And so we're really excited about it. It's really the first trial of its kind, and for some of these diseases, it's really the first disease-modifying trial. I just wanted to switch gears at the end. I was mostly talking about disease-modifying therapies, but it's still important to think about symptom management. And at the Memory and Aging Center, we really specialize in treating patients who have frontotemporal dementia, and this disorder, FTD, can actually be associated with a lot of really upsetting behavioral changes. Profound apathy, loss of empathy, social disinhibition, and it could really have a high caregiver burden associated with it. And we have some ideas about how to treat it, but it's still, we don't have any slam dunks. There's still not a great way to treat these symptoms. So oxytocin is an interesting hormone. So you may have heard about it as the hormone that promotes lactation. That is true, but it also promotes pro-social, kind of maternal, nurturing behaviors, increases social interest. And it's actually been tried in autism trials, and it actually has had some early success in promoting these pro-social behaviors in patients with autism. And so far in a phase 1, it was well tolerated in FTD. So we're actually currently running we're currently one of the sites for an oxytocin trial in frontotemporal dementia. We're really targeting social apathy. This is just a quick slide just to go over all the actively enrolling trials we currently have here. So we really have 2 immunotherapy trials, one in Alzheimer's, one in a broader range of tauopathies, one more anti-tau trial using a drug called sosylate in Alzheimer's disease, and the oxytocin trial. And we still have a lot more directions that we want to go. I can't advertise the other trials until they're cleared by our internal review board, but we still have a major interest in genetic frontotemporal dementia, particularly progranulin deficiency. I think we're going to be doing exciting things with that very, very soon. We still consider ourselves a major site of progressive supranuclear palsy research. I think we're going to be having more trials for that very soon. And we're always looking to increase our portfolio of Alzheimer's disease clinical trials. I think we're going to be doing a lot more exciting stuff with that soon. So feel free to reach out to us. Here's our contact information again. Thank you very much for coming for Braving the Rain. Thank you for the organizers. And I really wanted to thank our patients and their family members.